Okay, so we better make a start. First of all, thank you very much for joining us for this Ensembl release webinar. So my name is Denise and I work in the Ensembl outreach team at the EBI in Kingston, UK. I have muted all the microphones, but you're more than welcome to drop in any questions that you may have during this webinar. So if you want to address any question, please use the chat box and also make sure that you address the question to everyone so that all the participants of this webinar are able then to see what you guys are wondering about. So, and as I said, you are more than welcome as well to follow along the Ensembl-77.pdf file that is available for you to download. Okay, so this webinar is going to be just to give you guys some highlights on what's new in the latest release of Ensembl. So this latest release, release 77, version 77, was launched just a few weeks ago on the 2nd of October, and we've got exciting new things going on there in this new version of our database. So, for example, we've got a new version of the GENCODE genes for human. We also have got the very first merge between the automatic annotation and manual annotation for the rat genome. We also have got a few exciting things regarding assessing how reliable a transcript can be in human and mouse. So I'm going to be covering that in a few minutes. And also we've got a new species and a few other pieces of news on Ensembl 77. So let's first of all have a look at the GENCODE genes, version 21 in human. So the GENCODE is actually a consortium between 8 different institutions throughout Europe and the US. And the main goal of this consortium is to annotate all genes and transcripts based on biological evidence. So there are 8 groups out there working towards that target. And so what those 8 different groups are working on, so they are looking at manual and automatic annotations of human and mouse. They are also carrying out some experimental analysis and also some QC analysis as well. So this is just to highlight all the different institutions that are that take part of the consortium. So here at the EBI in Kingston, we've got Ensembl working towards the GENCODE, and also we've got the Sanger Institute where the Havana team are doing the manual annotation of those genes. So because we've got so many people collaborating together and using different types of analysis, so one can expect that the GENCODE is a version of annotation which has got a high accuracy and high quality. So therefore, the GENCODE, the set of genes annotated in human and mouse, they are the first choice for projects such as the ENCODE, the 1000 genomes, and also the International Consortium of Cancer. Cancer genomes. So that's the best choice of genes annotated out there. So it's being used by several different projects. So this is how the GENCODE pipeline would look like. So as I briefly mentioned, so we've got Ensembl performing the automatic annotation of all human and mouse genes. And we are collaborating very closely with the Havana team here at the Genome Campus at the Sanger Institute. So they contribute towards those genomes with the manual annotation of those genes and transcripts. And Ensembl is then doing the automatic part of this. So both teams are heavily relying on biological evidence. So we don't offer, we don't annotate our genes based on ab initio prediction. So we do have to have a piece of biological evidence backing up those annotations. So once we have our genes in human and mouse annotated by Ensembl and by Havana, this will then be fed into this AnnoTrack. So it's a tracking system that was developed by the Havana team at the Sanger Institute. And we also, oh, sorry for that. Yeah. We also then collaborate with some of the other groups, such as in CNIO, which is the Center of National Cancer Research in Madrid, and also with Yale University. So we are taking into account some of the predictions and also performing some experimental validation and verification of some of our transcripts. And in the end, once all this QC and the experimental validation are performed, we will then merge all this data, which will then give rise to the GENCODE data set. And this can be available in Ensembl, for example. So if you want to know more about this pipeline, I just recommend you guys to have a look at this publication over here, which provides more details. So for this new release of Ensembl, release 77, so those are the latest statistics on the human genome. So we've got the latest version of GENCODE, so it's version 10.1, and we've got just over 20,000 protein coding genes in human for this release. So you can access the GENCODE set of genes in human through our browser. So this is just to highlight how that view looks like on the Ensembl browser. So the data track is called as comprehensive gene annotation from GENCODE 21. So it's easy to follow and to find out which version of GENCODE we have in Ensembl. And you can also change the track style if you want to. So the GENCODE set, in addition to the browser view, can be also accessed through our tools. So we've got the data retrieval tool Biomart, we've got the variant effect predictor, and also you can carry out similarity search using our BLAST and BLAT. So this is all against the latest GENCODE set of human genes and transcripts. And in addition to the tools and in addition to the browser, you can also programmatically access the GENCODE 21 data via Perl, via MySQL, and also using our agnostic REST API. Okay, so now moving on to the second piece of news that I would like to share with you. It's regarding the rat genome. So for the very first time, we are now combining the automatic annotation by Ensembl with the manual annotation by the Havana team here at the Sanger. And so what you can see now in Ensembl, it's this merged gene set. And when the Ensembl automatic annotation overlaps and agrees with the manual annotation by the Havana team, we will color code that as gold. So if you are browsing the rat genome, you are going to be coming across now with golden transcripts and golden genes. And just remember that golden is good. So it's when we are confident that at least two different pipelines, Ensembl and Havana, automatic and manual annotation, they have agreed on the annotation of that gene or on that transcript. So this is already available in human, it's already available in mouse and zebrafish, and in Ensembl 77, this latest release of Ensembl, for the very first time we are able to provide this merge set for rat genes as well. And this has been welcomed very much by our user community out there. So as soon as we released that piece of news, so we had some people on Twitter, for example, pretty excited about having golden genes for rat as well. So how can you view those genes? So again, focusing on the browser, this is how the merge set would look like in red. So again, the golden genes here, they are a merge set between the automatic pipeline by Ensembl and the manual pipeline by Havana. You will also have genes that are annotated by either Havana only or Ensembl only, and they may not have been annotated by the two projects, so they will not be highlighted in red. So you can just click around here or there, and then this pop-up window will give you additional information, and you can find out whether it's a Havana only transcript or if it's a merge. So again, this is for Ensembl release 77. Now moving on to these two different flags or categories now that we are showing for human and mouse, which are called APPRIS and the transcription supported levels from the UCSC. So let's have a look what exactly they are. So you might be aware of, if we're working on human, mouse, rat, of how many alternatively spliced transcripts are out there. So we have talked a little bit about the GENCODE version 21, so we know that's the most comprehensive set of annotation out there. So we annotate every single transcript, providing there is biological evidence to support the splice junctions, the intron-exon splice junctions. So you may end up looking at a gene such as this one in human, the ATRX, and there are many different spliced isoforms there. So they are all alternatively spliced isoforms. So you may wonder, for your experiments, if you want to design, for example, primers, or if you want to perform additional analysis, which of those 19 or so transcripts you would focus on. So we are trying to help you guys out with this decision, the decision that you perhaps are facing on which of those 19 transcripts in the example of ATRX gene you would focus on. So we've got the CCDS annotation. So actually CCDS stands for Consensus Coding DNA Sequence. So it's an agreement between 4 different institutes, and we are highlighting to you the CDS that has been agreed between those 4 institutes. But now for this new release of Ensembl, we are also showing to you those flags, the APPRIS and the transcription supported levels flags. So again, this is another level that hopefully should help you out in finding a principal isoform, for example, that you perhaps would like to focus your analysis on. So those flags can be observed, for example, on the transcript tab on the browser view. So talking a little bit more about APPRIS. So this is a system that aims at annotating all splice isoforms and select a primary variant out of those possible isoforms there. So it's a system that has been developed by José Rodrigues, again at CNIO in Madrid. So the original paper was published last year. So what José and his colleagues are working on, or were working on, they were combining a set of different computation analyses, such as those 8 highlighted here. They also take into account conservation of residues and conservation of protein alignments. And then they are trying to highlight a possible splice isoform that could be tagged as a primary variant. So more information regarding the different computational methods that Jose and his colleagues have worked on. Just have a look at this URL here. But those different methods, they are just looking at different things. For example, they are looking for amino acid residues that may be of some functional importance. They also look at protein structure. And they also are looking at the conservation across several vertebrates there. And also they use this algorithm called inertia, and they are trying to also to take into account any information regarding selective pressure on the evolution of certain exons. So APPRIS have got this data. So they have run those 8 different computational methods for 4 different vertebrates out there. And they have called a principal isoform for those genes in those genomes. In Ensembl-77, we've got the data for mouse and human, and we are hoping to include APPRIS data for rat and zebrafish in the near future, probably in Ensembl-78. So just keep an eye for those genomes out there in our next release. So APPRIS will give you an idea which isoform might be the most relevant out of all possible alternatively spliced isoforms that GENCODE offers. You may also want to find out how well supported a given transcript model is. So if you look again at the browser, at the Ensembl browser, and if you turn on, for example, the EST alignments and the cDNA alignments, so you may recall that we rely on cDNAs, ESTs, and protein alignments to annotate our genes. So when you look at the browser, you will see that some of the ESTs and cDNAs, for example, they might be full length, so they might cover or encompass the full length of a given gene or a given transcript, whereas some regions of my gene will not be fully supported. So they will be supported by partial ESTs, such as this one here. So although we have some biological evidence that supports every single splice junction of a given transcript, we might have also partial cDNAs or partial ESTs. So that's when the TSL or transcript support levels created or developed by Mark Dickens at UCSC. So that's when the support levels come into play. So what Mark Dickens has done, they have started off with the GENCODE set. Remember the most comprehensive set of annotation for human and mouse. And he has carried out a pipeline, again, relying on alignments of cDNA and ESTs, assessing on the coverage of that annotation, whether it's fully covered or just partially covered by ESTs and cDNAs. And then they, Mark and his colleagues, have then designed those scoring levels and divided up the transcripts according to different categories. So in this next slide, I'm just highlighting the categories that you can come across with in Ensembl now. So they vary from 1 to 5. So the TSL1 or transcript support level 1 is the highest support level that one may wish to have. So at least one full mRNA sequence was able to support every single splice junction for a given transcript, all the way to transcript support level 5, where there is no much support out there, especially at the mRNA or cDNA level, and the only support comes from ESTs. You may also come across in Ensembl now with transcript supported level NA, which has not been analyzed so far. And the reason why a given transcript might not have been analyzed so far can be because it's a pseudogene. It can also be because that gene or transcript lies on an MHC regions such as HLL genes or immunoglobulin genes, or for those genes or transcripts that actually contains just a single exon. So those are things that Mark is still working on, and especially when it comes to single exon transcripts. So this will very soon be included in his analysis as well. So just a reminder, so when you are looking at those different TSL categories, so the highest supported one will be TSL1 all the way to TSL5, which is the most poorly supported category. Okay, so how can you view that information in Ensembl? So one of the ways, it's on the transcript tab. So you may come across now with those flags here. So we'll have things such as, again, this TSL1. So you may be pretty confident that there is at least one full mRNA that can support all splice junctions of this given Ensembl transcript. And according to APPRIS, this was classified as a candidate isoform. We also have other examples, such as this one, where the data from APPRIS judges that this transcript is actually very likely to be the principal isoform of this gene, and it's also in accordance with the TSL1 top level of being well supported. So again, this is brand new in Ensembl, but we do have already some help pages on both APPRIS and the TSLs in Ensembl. So just have a look at those if you start exploring Ensembl and you are not too sure what those new terms mean. Okay, so although we are trying to provide you guys with a set of different ways to assess how trustworthy a transcript can be, we would also like to reiterate that there is no single method out there that will tell us or will tell scientists and Ensembl users which transcript to use. So this is a decision that depends very much on a case-by-case basis. So we cannot, unfortunately, tell you which transcript you should use in your analysis or in your experiments. So some scientists might be looking at all transcripts, whether others will be happy just to look at one or two well-supported ones. So, but what we can do, we can try to help you out with the decision making by providing a list of choices. So you can look at whether or not there is a CCDS entry for that transcript. You can look to find out whether there is an agreement between the manual annotation by Havana and the automatic annotation by Ensembl. You can look at cross-reference entries as well. So there might be UniProt entries as well, RefSeq entries. So again, this can add another level of support for our genes and transcripts. And now the latest tools that Ensembl is providing are the APPRIS and the transcripts supported levels. So again, another level to help you guys out when making the decision. So I just want to now touch upon a few other news for this new release in Ensembl. So we also have got a brand new species. So it's the African green monkey, the Vervet monkey. So this is just a picture of all the 69 species that we now have in Ensembl-77, including this new primate. So in total, we've got 12 primate genomes in Ensembl. So the assembly for this Vervet monkey is the latest assembly dated from March 2014. So we are providing now in Ensembl the automatic annotation of genes of this Vervet genome, and as we speak, we've got just under 20,000 protein coding genes. And the importance of Vervet monkey comes across very much due to the fact that it's a well-studied model for biomedicine. So, for example, it's the most abundant host for the SIV. So again, it's a very much important species out there for biomedicine. So some of our primate communities are very much happy to have this genome and its annotation in Ensembl now. In terms of other news, so we have an improved gene set for Amazon Molly. So this is a quite exotic and weird genome that has been available in Ensembl for a couple of releases now, but in Ensembl-77, we have improved the gene set of Amazon Molly. So Amazon Molly, what is interesting about it is that it's an asexual vertebrate, so it's being used in studies for the persistence and evolution of sexual reproduction. So in Ensembl-77, we have done a manual assessment of the automatic gene models that we have generated. We have now got 23,000 protein coding genes, 3 more than the previous release of Ensembl. And we also have got 6, we've got RNA-Seq data for 6 different tissues. And if you want to know a little bit more about how weird or how interesting this species is, just have a look at our blog post here. So when it comes to RNA-Seq models, so this is what you can see now for Amazon Molly. So you need to configure the view in Ensembl, in the Ensembl browser, to show the RNA-Seq models there. So for Amazon Molly, we've got models based on RNA-Seq reads for 6 different tissues. And we also show here in red the annotation that we have done relying on vertebrate proteins. We also have got new alignment export icons in Ensembl. So if you are performing or if you are looking at either pairwise alignments or multiple alignments in Ensembl, you can now export what you see on the browser, and you can do so just by clicking on that icon over there, or there is also the button download alignment. So it's a different way of alignment, of exporting alignment from Ensembl, and there are different formats that you can export that alignment as, including the Mega and also Crustal W. Okay, so that's very much what I would like to highlight in terms of what is new for Ensembl-77. So I would also like to thank our users. Thank you guys for coming out and joining us in this webinar today. Especially, I would like to acknowledge my colleagues. So it's a huge group of very lovely people to work with, and we are all very much it's a big effort and a combined effort to provide this data to our users. And also would like to acknowledge and thank our funders as well for providing money for us to carry out this amazing work. So you can keep in touch with us if you are interested in finding out what would be our next webinars. I would strongly suggest you guys to follow us on Ensembl Twitter account at Ensembl. And you can also keep an eye on our Ensembl blog. We've got several tutorials on our tutorial pages. And again, you can always contact us on helpdesk@ensembl.org. So I think now we have time for some questions and comments. So are golden transcripts flagged in the database tables? Yes, they should be. So you could look for things flagged such as Ensembl_Havana. So those should be the golden transcripts. Is the agreement between Ensembl and Havana. So this should be available in the database tables, yes. Okay, so I think if there are no further questions, I'll just finish this webinar session then. Again, thank you very much for joining us and carrying on until the very end. I hope it was useful. And again, just keep an eye on our Twitter account to find out where the next webinar will be. And again, if there are any questions, just drop us a line at helpdesk@ensembl.org. Thank you very much again. See you later. Bye.